Hello, can somebody please confirm that you can hear me?
Yes, we can hear you.
Great. All right, are there any questions? Before I begin, let me state that I've done some grading. Let me go there and take a look. I know I've graded worksheet lecture 6. I think it was worksheet lab 7 that I just graded. This weekend, I'm going to try and grade quiz 2A and hopefully 3A. Any questions about any of that? All right, let's get going. How do I get out of here? There we go. I'll share my screen in a minute. I want to call up the syllabus. Uh-oh. I got the lecture. I must have moved it someplace. It's not where it's supposed to be. Oh, gosh. Where might I have moved it to? Now let me do a search. Let's look for chapter 8. No, that's searching the web. I don't want that. I'm going to have to go into the Clark. Hopefully it's still in Clark. It might have moved elsewhere. This is a little embarrassing. I should at least stop the recording here. I might have to wing this. Oh, wait. I know what I can do. We can go to here. And of course, I'm not sure where we left off. Did we finish chapter 6? Does anyone remember? I'm having a hard time finding.
The last note I have is DNA replication.
Is that in chapter 8?
I just have DNA replication as semi-conservative.
Yeah, that's chapter 8. Okay, we started chapter 8.
Yeah.
Now let me take a look at the syllabus here. Let me go share my screen now. The difficulty is that I normally have a lecture folder under Clark, and I'm not seeing the lecture folder. It probably got moved by accident. Let me see if I can do a search here. Does anyone know? Oh, here we go. Hopefully that can find that. Yeah, it looks like it's been moved to here. Let's see. D. Clark Fall 222. Yep, there it moved. All right. I'll move that later. Blow that up so I can see it. Get my better glasses on. I'm trying to. I guess I'm gonna have to stick with these. I don't see my other glasses. All right, so today is February 16th, and we're on chapter 8. And yeah, we will do a little bit of chapter 8 next week too. Let me see if I marked that where I was. Come on. All right, so we talked about DNA replication. And I think I showed you the video of this slide. So let's talk about DNA replication in more detail. This slide is showing you all of it, but we'll talk about it in a little more detail. DNA is copied by DNA polymerase. The new DNA, meaning the newly synthesized DNA, is made in the 5' to 3' direction. So on this picture, let's blow this up a little. On this strand of DNA, the DNA is made this way. This is the 5' end, this is the 3' end. So the DNA is made that way. That's true of this strand of DNA also. The trouble is in the lagging strand, this strand isn't 5' to 3'. And so the DNA is made in short batches this way, and this way. These short batches are called Okazaki fragments, after Dr. Okazaki, who discovered them. So when DNA is copied, DNA is not initially put down. An RNA primer is put down first, and then the DNA is synthesized on the RNA primer. That RNA primer, before the DNA replication is finished, is removed, and then the DNA is filled in. The leading strand is synthesized continuously because the DNA polymerase, once it binds to the leading strand, that's the template right here, the DNA polymerase can move directly straight down the DNA. And so this strand will be made continuously from whenever the DNA polymerase binds to whenever the DNA polymerase falls off. On the lagging strand, though, the DNA cannot be made continuously, and it's made in short batches, which I've already mentioned were Okazaki fragments. And the reason why it's doing that, is the DNA polymerase has to move this way on the strand, and the helicase, which was opening up the DNA, is moving that way. And so that's why we have Okazaki fragments on the lagging strand. Any questions about any of that? The RNA primers are removed. And the Okazaki fragments will be connected, and we'll talk about that, meaning they'll be removed, or the different pieces of Okazaki fragment have to be annealed together. And I'll talk about that in the next slide. I guess it wasn't that slide. How oh, there it is right there. Sorry, I just didn't see it. The Okazaki fragments are joined by DNA polymerase and DNA ligase. What DNA ligase does is wherever there's a discontinuation in the strand of DNA, the ligase ligates the two strands together. Like when, let's say this DNA polymerase falls off for whatever reason, and the helicase keeps moving this way, another DNA polymerase will bind and then will continue making the leading strand. Well, this new leading strand will not be connected to the, I guess not the leading strand, the replicated DNA will not be connected to the older replicated DNA. And DNA ligase will ligate together. Now, on the leading strand, the DNA ligase doesn't work very often. And it's possible that the DNA polymerase starts at the end of the chromosome and doesn't ever fall off. It can even replicate the DNA in one stretch. So there's only one replicated DNA. So the ligase doesn't have to do anything on the leading strand. Occasionally, though, the DNA polymerase falls off, and so there will be two strands of DNA, and then the ligase has to ligate them together. Does that make sense? So the leading strand doesn't use ligase that much, but it does occasionally. On the lagging strand, each of the Okazaki fragments has to be ligated together with ligase to its neighboring Okazaki fragment. So that means on this strand here, it would have to be ligated to this one and ligated to that one. And so the ligase is working quite a bit on the lagging strand. Any questions about any of that? Let me remind you of the three important enzymes in DNA replication. First, there's the helicase, which opens up the double-stranded DNA and makes it single-stranded. By single-stranded, the DNA polymerase can then come in and then replicate the DNA. So the second enzyme is DNA polymerase. And then the third enzyme that's of critical importance is ligase. There are other enzymes involved, but those are the three critical ones you should know about. Any question about any of that? All right, all of the DNA is synthesized to make the newly replicated DNA. And that is DNA replication. So now let's talk about RNA transcription. The synthesis of RNA from DNA is called transcription. DNA is transcribed to make RNA. And which RNA would be transcribed? Would it be messenger RNA, tRNA, or rRNA? Anyone have a guess? Anyone have knowledge? It's really not that difficult.
Messenger RNA?
Most of the genes will be transcribed as messenger RNA because most of the genes code for a protein. However, not all of the genes will be transcribed as messenger RNA because some of the genes code for tRNA, and they will be transcribed as tRNA. And some of the genes code for rRNA, and they will be transcribed as rRNA. But about 95% of the genes, certainly over 90% of the genes, code for protein, and they will be transcribed as messenger RNA. Okay, any question about that? And I think it's actually over 95% of the genes are transcribed as messenger RNA, because there's only a handful of tRNA genes. I think there's only a handful of rRNA genes as well. Certainly there's less than 24 of each. And then that tells you that not all of the DNA is transcribed. Only the genes are transcribed. Any question about that? Now, in eukaryotes, there is a lot of DNA which does not code for a gene. In fact, most of the DNA is non-coding DNA in a eukaryote. In prokaryotes, most of the DNA does code for a gene. And I guess I should say in bacteria, not in prokaryotes, because we're talking about bacteria here. And archaea are a little different than bacteria, at least in this regard. I think most of archaea does code for a gene, but there definitely is non-coding regions in archaea. And bacteria doesn't have much non-coding DNA. It does have some, but not much. DNA transcription begins when the RNA polymerase binds to DNA. And the RNA polymerase binds at one site in the DNA, and that site is called the promoter. Any question about any of that? The promoter is a region of DNA upstream of the gene. And then the RNA polymerase moves down the DNA to transcribe the gene. Transcription also proceeds in the direction 5' to 3', so it's the same direction as DNA replication. And then RNA is made in the 5' to 3' direction. The RNA polymerase continues transcribing the DNA until the RNA polymerase reaches a region of the DNA called the terminator. The terminator is a region of DNA after a gene, and the terminator codes for the RNA polymerase to fall off of the DNA. When the RNA polymerase falls off the DNA, it's obviously released, and that also releases the RNA that has been transcribed. Any question about any of that? All right, remember the non-coding DNA is not transcribed. And while we're talking about the information in DNA, meaning the information that's coded, that information will be transcribed into RNA. So the information is passed from the DNA to the RNA. Any questions about any of that? All right, this is an actual, it's not exactly a electron microscopic image, but it's similar to that. It's using fluorescence microscopy to look at RNA transcription. And there's the RNA polymerase. I think right there is the RNA being made. And that's the strand of DNA that the RNA polymerase is moving down. Let's talk about RNA transcription in a little more detail. I don't think this link works. I think I got to go to YouTube to get that. Yep, it's not available anymore. Oh, take that back. It was available.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that? There's no questions, we'll move on. Let me close that down. This model is showing you that the RNA polymerase begins by binding to the DNA at the promoter. And then the RNA polymerase moves down the DNA and begins transcribing the DNA to make RNA. Any questions about any of that? The transcription of RNA continues until the RNA polymerase reaches the terminator. So that should be specified as the promoter up here, and then the terminators here. The gene is the region in between the promoter and the terminator. It is true there might be something right after the promoter, but the terminator always is at the end of the gene. So it's after the gene, and it codes for the RNA polymerase to fall off the DNA, and that will release the RNA. So then it can move into the cytoplasm. And I guess that's what that slide's showing you. Once the RNA moves into the cytoplasm, we can then have protein translation occur. Protein translation requires 3 pieces of RNA: ribosomal RNA, messenger RNA, and tRNA. Okay, now the ribosomal RNA is in the ribosome, and the ribosome has both ribosomal RNA, or rRNA, and ribosomal protein. So both the protein and the rRNA make up the ribosomes. Any question about any of that? All right. So the messenger RNA brings the information that's in the gene to the cytoplasm to be translated into protein. So the messenger RNA has the information needed to make the protein. The ribosomal RNA and the ribosomal protein making the ribosome puts it all together, and that's where the protein is made, at the ribosome. The tRNA is playing a unique role, and it brings in each amino acid to make the growing protein. And it's the tRNA which is reading the code in the messenger RNA. Any questions about any of that? So messenger RNA is translated in groups of 3 nucleotides in the messenger RNA. And we call these groups of 3 nucleotides codons. So one codon codes for an amino acid. And a codon is simply 3 nucleotides in the messenger RNA. Surprisingly, when the ribosome moves down the messenger RNA, it does not initially begin translation of the messenger RNA into protein. The ribosome has to move down the RNA till it reaches the start codon, which is AUG. And this one you should probably know. AUG is where the ribosome will start protein translation on the messenger RNA. And AUG also tells the ribosome to add the first amino acid, which is always methionine. So AUG plays 2 functions. It codes for the amino acid methionine, and it also tells the ribosome, begin protein translation here. Now, later, when the ribosome moves down the messenger RNA and it runs into AUG, usually the AUG just codes for the amino acid methionine. Okay? And I don't really want to talk about what it could do else. And it does allow the ribosome to make a truncated protein, meaning if the first AUG wasn't made or the ribosome binds to the messenger RNA after the first AUG, it can then start translation at the second AUG, and you don't need to know that because we're not going to talk about that. I was just filling in that most of the time the AUG just codes for, the second time, it just codes for the amino acid methionine. So know that the AUG codes for the start codon and it codes for the amino acid methionine. Now, when the ribosome moves down the messenger RNA, it will continue adding amino acids for each codon up until the ribosome reaches a codon that codes for stop. And you don't need to memorize these, but UAA, UAG, and UGA code for stop. And when the ribosome reaches the stop codon, it stops protein translation. Any question about any of that? All right. So in overview, the DNA, the information in the DNA, when the information is in a gene, is transcribed to messenger RNA. That information in the messenger RNA is then translated into protein. Any questions about any of that? All right. So when we're talking about translation, and we mentioned that 3 nucleotides is a codon, and each codon codes for one amino acid, we run into a problem because organisms contain about 20 different amino acids. Most organisms have 20 different amino acids. Humans have 20. I think E. coli also has 20. The mouse might be 22 or 23, and we're not going to talk about any of those. But most organisms have 20, but yet there's a possibility of slightly over 60 possible codons. So what happens to the rest of the codons if only 20 of them code for an amino acid, or there's only 20 amino acids? What happens is there's more than one codon for most of the amino acids. I think there's two that only have one codon. The first is AUG, coding for the amino acid methionine. And then there's one other, there it is there, tryptophan is only coded by UGG. All the others have more than one codon code for that amino acid. Some of them, such as phenylalanine, have two codons that code for that amino acid. Others, like serine, have 4 amino acids coding for that, not codon, but that amino acid. And then some, like leucine, have 6 codons coding for that amino acid. You don't need to know the numbers, and you don't need to memorize the table. Like I said, the only codon you probably should know is AUG, coding for both methionine and the start of protein translation. So the table of the codons shows you that there is redundancy in the codons, that one amino acid can be coded by more than one codon. And that's true for most of the amino acids. And this is called the degeneracy of the code. Degenerate in the fact that there's more than one codon for each amino acid, with the exception of the 2 that I mentioned. Any questions about that? All right. So let's take a look at a translation video. Turn it down at the beginning.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about that? No questions? Boy, I hope I didn't lose that. All right, let's talk about protein translation in a little greater detail. Here we see the overview of protein translation. Protein translation requires the ribosomes, messenger RNA, and tRNA. The messenger RNA encodes the codons, and the tRNA actually reads the codons. And how that happens is the tRNA has an anticodon, which actually hydrogen bonds to the codon, meaning A will hydrogen bond to U, U will hydrogen bond to A, G will hydrogen bond to C. And by reading this codon, the tRNA brings in the correct amino acid. Any questions about that? The ribosome puts everything together and then moves down the messenger RNA. The ribosome has two sites in it that allow tRNA to come in and then bind to the messenger RNA. And so the ribosome is putting everything together and, I don't know, moving down the RNA. First, the ribosome will move down the messenger RNA and won't do anything until it reaches the start codon, which is AUG. Once it reaches AUG, it will allow the first tRNA to come in, and that will be the tRNA carrying the methionine amino acid, and it'll have the anticodon UAC, which you don't really need to know the anticodon. In fact, you can figure it out, because if you know the start codon is AUG, the anticodon has to be UAC. Any questions about any of that? All right. Well, the ribosome has a site for a second tRNA to come in, and the tRNA that will come in will be the one that has the anticodon AAU because that's complementary to UUA. And that UUA is not always the second codon, it's just the second codon that we're giving you an example to. And that will bring in the second amino acid. Okay. Now let's see. What happens next is the methionine is removed from the first tRNA, and that methionine is added to the amino acid on the second tRNA, which in this case is leucine. So methionine will be added to leucine. And your book doesn't discuss this, but the enzyme that is removing the methionine from the first tRNA, or the tRNA in this position, and then adding it to the newly added amino acid, that enzyme is actually a rRNA enzyme. And this is the only rRNA enzyme, the only RNA enzyme that we're going to discuss. Okay? Any questions about that? So the only enzyme, which is an RNA enzyme we're going to discuss, removes an amino acid from the tRNA and then adds it to the newly added amino acid. You don't need to know which, this is called the P site and not the A site. You don't need to know that. Once the amino acid is removed from the tRNA, the tRNA is free to leave the ribosome. That will then allow the ribosome to move down one codon. And I don't know how this works, but this tRNA and this amino acid now move to the first position. And it's not that they get out and move and go here. It's that the ribosome moves down. And like I said, I don't know how that works, but the second tRNA is now in the first position of the ribosome, and that means this frees up this position of the ribosome so that the next amino acid can come in. And when it comes in, it'll have the anticodon for the complementary to the codon down here, and it'll bring in the correct amino acid. And then the leucine, or the second amino acid, is removed from its tRNA. So that's broken here. And then it is added to the newly added amino acid. And now we're going to have a growing protein of 3 amino acids. Okay. That will free up this tRNA to come out, shown there. That will cause the ribosome to move down one codon. The last amino acid, CCA, if that's shown. Actually, it's not shown here. This one then moves to the first position, and that allows the next tRNA to come into the second position of the ribosome, which, let me see where that is. So this one has already done that and moved, and it's showing you right here. And this is how the amino acids get added to the growing polypeptide that when it finishes, we'll call it the protein. So the finished product here will be the protein. The ribosome just keeps moving down the codons allowing another tRNA to come in, which will then be added. That one is actually methionine being added here, which will then add another amino acid to the growing polypeptide, which will eventually become the protein. That continues until the ribosome reaches a stop codon. When the ribosome reaches a stop codon, it codes for protein translation to N. The ribosome will then disassemble and fall off the messenger RNA, and that will release the newly replicated protein. Any questions about any of that? I had a question for you. Let me see if I can remember what it was. I just had it. Oh, what happens with a protein that we don't want methionine to start the protein? Anyone have a guess? Because all proteins start with methionine when they're being translated, but not all proteins want to start with methionine. So some of them don't start with methionine. And how do they get rid of the methionine? Come on, guys, it's not that difficult. All right, think about that. So here we're looking at a complication of protein translation. And that is showing you, I wonder if I should even start talking about that one. We'll start talking about that one. It's showing you that that is actually the DNA. And these are the messenger RNAs being made on the DNA. And then this is showing you that protein translation is occurring on the messenger RNA, which is being made. So in prokaryotes, we have a complication, and that is the RNA is being transcribed in the cytoplasm because there is no nucleus. And as you know, protein translation occurs in the cytoplasm of both eukaryotes and prokaryotes. And in prokaryotes, we can have more than one messenger RNA being made from the same gene at the same time. That's why this one is longer. The messenger RNA is close to being finished, if not finished. And then it gets shorter and shorter and shorter and shorter. And this one, the messenger RNA, is just starting to be made. So that's the first complication, and that is in prokaryotes, and this happens in eukaryotes too, you can have more than one messenger RNA being made on the gene at the same time. In prokaryotes, it's even more complicated, and it's showing it best here. It is showing it here, but it's hard to see there. And that is in prokaryotes, on the messenger RNA, we can have protein translation begin even before the messenger RNA is finished being made. And so these red, I don't know, hearts or something like that are showing you that protein translation is being made. And I guess they're showing you the amino acids. And so this one is a longer polypeptide than that one. It's longer than that one and longer than that one. And that's because this messenger RNA has just recently been made. That one was made a while ago. And that's why the messenger RNA is longer here than here. And then the proteins being translated there's more of them here because this messenger RNA has been around longer than this messenger RNA, which has fewer amino acids on it. The point is, in prokaryotes, there's a complication, and that is you can have more than one messenger RNA being made from the same gene at the same time. And while the messenger RNA is being made, you can have protein translation starting before the messenger RNA is finished. Any questions about that complication? And that's what this slide is trying to show you, the complication. Now, in eukaryotes, we never have protein translation occurring before the messenger RNA is finished being transcribed, because transcription occurs in the nucleus and translation occurs in the cytoplasm. So the messenger RNA has to move out of the nucleus and move into the cytoplasm before protein translation can begin. Any question about any of that? All right, so that's the complication of what's going on in prokaryotes. The complication in eukaryotes is a little different. And there's two complications in eukaryotes. In eukaryotes, first, you can have a gene which has regions of the gene, meaning parts of the DNA, that actually do not code for the protein. And then regions of the DNA portion of the gene which code for the protein. The regions of the gene that code for a protein are called exons. And then the region of a gene that does not code for a protein are called introns. Bacteria do not have introns in their genes. They only have exons, meaning all of the region of a bacterial gene codes for protein. But in eukaryotes, there's a lot of the gene which does not code for the protein, and that we call introns. When the messenger RNA is first made from the gene, the introns will be transcribed into that initial RNA transcript. This initial RNA transcript cannot leave the nucleus. There is a check on the pores of the nuclear membrane, and it apparently checks the RNA and makes sure there's no introns. If there's an intron, the messenger RNA cannot get through the nuclear membrane. What happens with this initial transcript is that the introns are cut out, and that's called spliced. And then the exons are annealed together, which is also called spliced. They're spliced together. So it's called splicing out the introns and then splicing together the exons. And then only the final messenger RNA that contains only exons is allowed out of the pore of the nuclear membrane. And of course, this will be the messenger RNA, which will be translated into protein. Any questions about any of that? All right, so there's a complication of transcription and translation in prokaryotes, meaning there can be more than one messenger RNA being transcribed, and then translation occurs in prokaryotes before transcription ends. In eukaryotes, there's another complication, and that is eukaryotes have introns in their genes, and the introns have to be removed from the messenger RNA before that messenger RNA will be allowed to leave the nucleus. All right. If there's no questions about that, let's take a look and see where we've gone. We've talked about the flow of genetic information, DNA replication, transcription, and translation. Let's move into the region of the regulation of bacterial genes called gene expression. And to do that, we need to talk about operons. We probably won't talk about mutations today. So when a cell is regulating its contents, one way to regulate the contents in a cell are to control gene expression. Some genes are expressed at a fixed rate, such as a constitutive enzyme, an enzyme which is always used by the cell and it's always made at a fixed rate. Other genes are expressed only as needed, and these are repressible genes and inducible genes. And of course, a gene can code for an enzyme, and so there are repressible enzymes and inducible enzymes. There are other ways to control the products in a cell. You can regulate the cell by pre-transcriptional control, and that's what we're going to talk about, the regulation of gene expression. It uses operons, and we're only going to talk about inducible operons. There's a repressible operon, which is very similar, but we're not going to cover it. We're not going to test. I'm not going to test you on it. Another way to control a gene is by epigenetic control, and I'm not going to even talk about that. It's really kind of hairy, and I don't think your book even talks much about it. It's kind of complicated, epigenetic control. And then, of course, there's post-transcriptional control, which we're not going to talk about, and you're not going to be tested on it. But the enzyme can be controlled in other ways, whether the gene is turned on or turned off. You can make the gene, for example, and then inactivate the gene. You could also make the gene, meaning make the enzyme, and degrade it. That's another way to control what's inside the cell. All right, so we're going to talk about inducible genes, and let me begin by saying, if a gene isn't going to be needed by the cell, does it make much sense for the cell to make the product if it's not going to be needed? And the answer is no. Why bother making a product if the cell is not going to make it? And if the cell is not going to use it. If that's the case, we then have inducible operons, or maybe a repressible operon, to control the gene expression. If it's not needed, why have the gene turned on? It would be much simpler and much more energy efficient just to not express the gene, meaning not transcribe it, not translate it. Okay, to do this and to talk about gene expression, we need to talk about operons. A bacterial operon contains a gene of interest, which is usually called a structural gene. And usually when we're talking about a gene, we're talking about a structural gene. It's just that I've never used the term until now. For example, I think I've said, I've talked about the blue-eye gene. That is a structural gene. We've talked about the gene for encoding the enzyme catalase. That is a structural gene. We talked about the gene for encoding hemoglobin. That is a structural gene. So most of the times when we're talking about a gene, we're meaning the structural gene. However, there are regions around that structural gene that, I don't know, control the expression of that gene. That's one way to word it. You've actually been introduced to two regions already, and that is the terminator is a region of DNA after the gene that stops protein transcription. And then we talked about the promoter. We are going to talk about the promoter in much greater detail now, and then talk about other regions of the DNA that control the expression of the structural gene. Eukaryotes are similar to this operon, except they're a little different, and that is bacterial operons may have one structural gene on the operon, but they may have more than one structural gene on the operon. And if there's more than one structural gene on the operon, they will be related genes. For example, in this case, the Z gene encodes the enzyme for the sugar, digesting the sugar lactose, and so that's the lactase enzyme. The Y gene codes for the protein which brings the sugar lactose across the cell membrane of the cell. And then the A gene is involved in the metabolism of lactose. Once you divide lactose into monomers, you get beta glucose, glucose and beta galactose. And the A gene is involved in converting or helping to convert a beta galactose into the sugar glucose. And you don't need to know why that happens, but glucose is the base sugar, and we've talked about it. Glucose is the preferred starting point for like aerobic respiration and fermentation for that matter. So generally the other sugars are just converted into glucose. And the A gene helps convert beta galactose into the sugar glucose. The point is all the structural genes are involved in the metabolism of the sugar lactose. And prokaryotic operons are that way. Eukaryotic operons are similar. However, they only have one structural gene to each operon. And in humans, we do have the enzyme for lactase. We do have the Y gene coding for the protein to bring in the sugar lactose across the cell membrane. And then we have the A gene for converting beta galactose into the sugar glucose. So instead of having one operon, eukaryotes would have three, because they would have one for the Z gene, structural gene, one for the Y structural gene, one for the A structural gene. Any questions about any of that? All right. So an operon includes a structural gene or genes that are related to each other. And then the converting regions around that structural gene that help coordinate the expression of the structural gene. These include the promoter and the operator, and it's not shown here, but I think it does include the terminator. Any questions about any of that? All right. So how is the lactose gene or lactase gene controlled? Control can be by induction or repression. The lac operon is an inducible operon, And that means that the gene is normally turned off. The lactase gene is normally turned off by a prokaryotic cell. And that's because cells are not likely to be in an environment around the sugar lactose. If you don't know, that sugar is found in dairy products, and that's about it. It's not a very common sugar. So unless the bacteria were on a dairy farm, the bacteria is not likely to see the sugar lactose. So the bacteria, such as E. coli, shuts down the lactase gene normally, meaning most of the time the lactase gene is shut off, and that is an inducible gene. It can be turned on when the cell wants to turn it on. We'll talk about that in the next slide, which I don't know if I'll make it or not. How the Z gene, the lactase gene, is shut down, and when the Z gene is shut down, all of the members of the lac operon are shut down, meaning all the other structural genes in the operon are shut down as well. How the gene is shut down is by another gene. And this is a regulatory gene. It's called the I gene. It makes a repressor protein. The repressor protein can actually bind to the operon and then shut it down. Okay. Now, in the case of the lac operon, the gene that regulates the lac operon is right next door to the lac operon, so that the regulator is right next door to the gene that it operates on. Most of the time, that is not the case. The regulatory gene is found somewhere else in the bacterial chromosome. And in eukaryotes, the regulatory gene is actually found usually on a different chromosome than the gene it's regulating. Any questions about any of that? But in the case of the lac operon, the regulatory gene is right next door to the gene it regulates. That, like I said, is not normally the case. How the regulatory gene works is it makes a repressor protein. That repressor protein can bind to the operator, which is a region of the operon, right here, the O region, and the O region follows the promoter region. So when the the RNA polymerase binds to the DNA and then begins moving down the DNA to begin RNA transcription, the RNA polymerase will be stopped by the repressor protein, and it will not move further. So that the Z gene, as well as the Y and the A, are not going to be transcribed. This effectively shuts off the gene. And we are going to be talking about gene expression in terms of the gene is turned on or the gene is turned off. And this is a simplification just to be easy to discuss it. In reality, the gene can be turned off, but it can be at various levels of being turned on. So there could be very few copies that are transcribed of the gene, or there could be some copies of the gene that are transcribed, or there can be a lot of copies of the gene transcribed, or there could be many, many copies of the transcribed gene, meaning the RNA is transcribed. And we're not going to talk about the complication. We're just going to talk in simple terms. The gene is either off or it's on. If the repressor protein binds to the operator, the lac operon is turned off. Any question about any of that? And that's because the RNA polymerase cannot move past the operator once the repressor protein is bound to it. So, is that polymerase going to be stuck there forever? Or do you think it's going to fall off?
It'll fall off.
Yeah, eventually that polymerase will fall off. The polymerase binds to the DNA by a stoichiomic reaction, and every once in a while it will fall off, in which case it can bind to another promoter and then transcribe that gene. It probably won't bind to this promoter again. It's most likely going to bind to another one, but it could bind to this promoter, in which case it'll move down to the repressor protein and then stop moving. Okay. All right, so that explains how the gene is turned off. And we mentioned that E. coli normally wants the lactase gene turned off because E. coli is not normally going to see the sugar lactose. And so it doesn't want to make the enzymes needed to digest the sugar lactose if there is no lactose around. But how do we turn the gene on? You actually turn the gene on by an inducer. And in the case of the lac operon, the inducer is itself the sugar lactose. Now, in reality, there is the sugar lactose, which is right-handed or left-handed as an isomer. And I don't remember if it's right-handed or left-handed. I'll go look it up. And allolactose is the same sugar, but it's of the opposite hand. So if one is the left hand, the other is the right hand. And they're just the same sugar, but they're different isomers of that sugar. And hopefully you know what I'm talking about, because I don't want to talk about what an isomer is. Okay, but I kind of mentioned it. It's the same molecule. There's just one that's right-handed, or excuse me, right-handed. There's one that's left-handed. And I don't remember which one is which. I'd have to look it up. I used to know in my younger days that you normally see sugars as one, right-handed or left-handed, and I don't remember, so doesn't matter. The point is that whenever the sugar lactose is around, you have the sugar allolactose around because they're the same sugar, just isomers of each other. Whenever allolactose is around, it can bind to the repressor protein and then inactivate it. If the repressor protein is inactivated, it cannot bind to the operator. So if all of the repressor protein is bound to the inducer, which in this case is allolactose, then none of the repressor protein can bind to the operator. And then the gene will be turned on, because when the DNA polymerase binds to the promoter, it'll remove right down through the operator and then start transcribing the Z, the Y, and the A genes. And they're structural genes. Any question about that? Well, here's a question for you. What happens to the repressor protein that's already bound to the operator? And I'll give you a hint. If it's bound to the operator, allolactose cannot bind to the repressor protein. So how do we get that off the DNA so that we can turn the gene on? Anyone have a guess? It's not that difficult. I'm running out of time, so let me put in the answer. And that is the repressor protein doesn't permanently bind to the operator. It's binding as a stoichiomic reaction, and every once in a while it will fall off. Now, in the case where there's no allolactose around, there will be active repressor protein, and it will bind to the operator, and that will keep the gene turned off, meaning this will fall off. There's no polymerase here. This will bind, or this one will bind to it again, and then the gene will continue to be off. However, if there's allolactose around and this repressor protein falls off, the allolactose will bind to it and then inactivate it. And that will cause the operator to be free, and then the transcription of the Z, the Y, and the A genes will occur. Any question about any of that? All right, when we make this messenger RNA, what's a little different about it? Shown right here. How many proteins do we get from this messenger RNA?
Is it 3?
Yeah, it's 3. That's a little unusual. All the other cases we've talked about a messenger RNA, you only made 1 protein from it. In the case of the bacterial operons, though, if there's more than 1 structural gene on the operon, you will make more than 1 protein. In the case of the lac operon, this messenger RNA will be translated into 3 different sugars. Beta galactosidase is another name for lactase, the enzyme lactase. And so that'll digest the sugar lactose. A permease is the protein that brings the sugar lactose into the cell, across the cell membrane. And then transacetylase, I already mentioned, is the enzyme that helps convert beta galactose to the sugar glucose. So all 3 of the structural genes are involved in the metabolism of the sugar lactose. All right, let me see. I think that's about it. Well, we've got a lot to go, so let me go ahead and at least talk about one more slide. And we're going to move on from the lac operon and operons and begin talking about mutations. A mutation is a change in the genetic material. So the DNA in its sequence gets changed, and that will be a mutation. Mutations may be neutral, meaning they give no benefit to the individual having the mutation, but no harm. They can be beneficial, in which case they benefit the individual. I can give you one example of one. Or they can be harmful. And I think you know of some harmful mutations, like what the heck is that called? Hemoglobin, when the hemoglobin is made differently in sickle cell anemia, that would be a harmful mutation. Okay. Beneficial mutation, it doesn't happen all that often, but every once in a while it can happen. There was a case where somebody was growing fruit flies in a glass container. I'll make it like this. And they had a cotton plug on top, and the fruit flies would be kept in the jar, and there'd be food at the bottom for them to eat. And the pupa would come up on the jar, and then they would form in their pupa. And when the jar got very crowded, there wasn't any room for more pupa. So there was on the side of the jar. So there was a mutation in the fruit flies, which allowed them to put their pupa on the cotton, which was the plug of the jar, having the food and the fruit flies in it. And so that was a beneficial mutation, because now the fruit fly could have an offspring, and what happens is you have the larva, which then goes through the pupa stage. And the larva could find some place to become a pupa. Whereas if it didn't have that mutation, the larva could not find any place to have the pupa. And so that mutation was beneficial and it increased in frequency in the population. There was another mutation in the human population, which we think when it came around was actually a beneficial mutation, and that was the blue-eye gene. It happened in Northern Europe about 10,000 years ago. Before that time, humans only had brown eyes. And they think back then, for some reason, the blue-eye gene was beneficial because it increased in frequency. In fact, in Northern Europe nowadays, the blue-eye gene is more common than the brown-eyed gene. Today, there does not seem to be any benefit of having a blue-eye gene. So we'd say it is now a neutral mutation. But back when it happened 10,000 years ago, there may have been a benefit of having blue eyes. What it was, we don't know, and why, we don't know. A mutagen is an agent that causes mutations, and then spontaneous mutations can occur without a mutagen, and that's just where an accident happens when DNA is being replicated, and that we call a spontaneous mutation. Any questions about any of this? If not, it's time for me to end, and I will see you at 6:30 for the lab. Bye.